And it is my great pleasure to welcome today Katrin Preller from the University of Zurich, who is going to speak today about the neurobiological mechanisms of psychedelic drugs. So Katrin Preller initially got a master's degree from the University of Constance about neuropsychology and clinical psychology. Then she joined for her PhD at the University of Zurich, where she started to investigate long-term effects of cocaine, MDMA, and heroin use. And after she completed her PhD, she received a Swiss National Fund postdoc mobility fellowship, with which she was going to London to join the at UCL, and in Yale University, where she is also still holding a visiting assistant professorship. But she is now located with her own junior group as a junior group leader at the University of Zurich, with a focus on neuroimaging with pharmacological challenges of the participants. So I'm very excited on the one hand about the methodological, very interesting challenges which come with pharmacological FMI, and on the other hand on the topic which we have today here in like this Christmas lecture, like the last lecture before Christmas, like the colorful world of psychedelics with neuroimaging. And I'm very much looking forward to your talk, Katrin Preller.
Timo, thank you so much for the nice introduction, and thank you for the invitation. I'm really excited to be here today, and deeply regret that I missed to put in some Christmas pictures into the slides. I hope they will still be colorful enough though. But give me just a second to share the slides. Okay. So so as Timo already introduced, I'm going to talk about the neurobiology of psychedelic substances. I hope I planned enough time to have a discussion afterwards. If there's anything pressing, feel free to interrupt me anytime. But you might have to speak up because I'm not able to see everyone right now. So just interrupt me. And also I planned for at least 20 minutes of discussion at the end of the talk. Okay, so the neurobiology of psychedelic substances. Well, psychedelics we know are inducing an altered state of consciousness that is characterized by visual alterations, by alterations in emotion processing, by alterations in how we perceive our body. But why would we think that the neurobiology of these substances is interesting? Well, first of all, we think that psychedelics can tell us a lot about how the brain works in health as well as in disease. And the other major reason why we are interested in that is because there is evidence, and it's accumulating right now, that psychedelics may be beneficial in the treatment of various psychiatric disorders. And of course, we kind of want to know how they do that. Why are they effective in treating psychiatric disorders? So the two substances that I've worked with the most during the last about 10 years are psilocybin and LSD. And they share an overlapping phenomenology, meaning they're inducing pretty similar effects that our participants are reporting on. However, their pharmacology is slightly different. So psilocybin is a preferential serotonin 2A and 1A agonist. LSD, on the other hand, stimulates various serotonin as well as dopamine receptors. So mentioning the subjective, the phenomenology, the subjective effects, as I said, we have various alterations of sensory perception, which I'm not sure if you can see my pointer here, but so visual alterations, sensory alterations are very, very prominent, which is also the reason why they have been called hallucinogens. When we prefer the term psychedelics now, because even though these visual alterations are prominent, it's not the only thing that they're inducing. So we're also talking about, as I said, disembodiment, feelings of disembodiment, changes in the meaning of percepts, insightfulness, some experience spiritual experiences, or the experience of unity. Now, the reason why I'm showing this in so great detail, even though we're more talking about the neurobiology, is because even these purely self-reported effects already tell us a lot about the pharmacology of these substances. And this is two different studies, one that we've run with psilocybin and one that we've run with LSD. And in these studies, we did not only administer psilocybin and LSD, we also pre-treated our participants with Ketanserin. Now, Ketanserin is a fairly selective, it's not perfect, but fairly selective serotonin 2A receptor antagonist, meaning that it blocks the serotonin 2A receptor. And when we then later administer psilocybin or LSD, the substances reach all the other receptors that they're targeting, except for the serotonin 2A receptor. And here's basically, I mean, the picture is basically the same. So psilocybin itself, the blue bars here, induce this altered state of consciousness that we were just talking about. LSD here in purple does exactly the same, altered state of consciousness. However, when we administer Ketanserin, we see the green bars here that they're pretty much exactly the same as placebo, which is black here, so barely noticeable. And the same when we look at LSD, green and yellow bars, pretty much the same thing. So meaning when we block the serotonin 2A receptor, our participants cannot distinguish anymore whether they had LSD or psilocybin or placebo, which is quite remarkable because these are really powerful psychoactive substances. So this already tells us that the serotonin 2A receptor is key in inducing the psychedelic effects of these substances. But that still doesn't answer the question, how do we get from stimulation of the serotonin 2A receptor to these altered states of consciousness that our participants are describing? And how do we get from there to the clinical effects that we see in these preliminary clinical trials that people are conducting right now? So for the talk today, I'm not going to talk too much about this last bit. We are currently conducting 2 clinical trials, and I hope when we meet again, hopefully sometime next year, I can present some results on the insights derived from these clinical trials. For today, I will focus mostly on how to get from the receptors to the altered state of consciousness, and just at the very last bit of the talk about some of the hypotheses that we have from how we get from those to the clinical mechanism of action. So we know that the serotonin 2A receptor is an excitatory receptor. And that means once our psychedelic enters the brain and stimulates the serotonin 2A receptor, the cell has a higher likelihood of becoming active or firing. Now, this, of course, depends on the specific brain region, because in some brain regions, there are more serotonin 2A receptors than in other. That being said, serotonin 2A receptors are distributed across the whole brain. So it's literally everywhere, but it's more prominent in some areas than in others. Then, of course, this depends on the exact dose that was administered, and it depends on the substance that was administered. As I said in the beginning, even though there are overlapping effects, the pharmacology is slightly different from one substance to the other. And even though we know that serotonin 2A is key in producing these effects, that doesn't mean that the other receptors which are being targeted don't also have some kind of modulating effects. So the next part of my talk is basically trying to answer the questions: how do we get from changes in neuronal activity to changes in information processing across the whole brain? And given that our brain is not an island, but it's interacting with our body and our body is interacting with the environment, is the brain, or how is the brain processing this interaction with the environment under the influence of psychedelics. So starting with this first part, what's happening on a system level across the brain under the influence of a psychedelic. We started out investigating a pretty old theory, which was put forward by Mark Geyer and Franz Vollenweider. So what these two suggested based on animal studies was that the thalamus, so this big structure here in the middle of the brain, is key in producing the effects of a psychedelic. And what they suggested is that the thalamus usually filters the information that we receive from either within ourselves or from our environment, and that this filtering function is not working quite as well under the influence of a psychedelic, meaning that more information is being passed through the thalamus to the cortex, which leads to sensory overload, which is then eventually producing these psychedelic effects. So we wanted to look at what exactly happens when we're thinking about thalamic connections to the rest of the brain. And we first did that by taking a seed-based approach and looked at the thalamic seed and the connectivity with the rest of the brain. And as you can see here very nicely, we see that LSD, this is LSD data, LSD is changing the connectivity between the cortex and the thalamus with increased connectivity between the thalamus and sensory brain regions. So for example, the occipital cortex here, we see that, so all the bright or the warm colors are higher connectivity, all the blue colors are lower connectivity. So increased connectivity with sensory or sensory motor brain regions, decreased connectivity with association brain regions. But as you of course all know, seed-based connectivity is a correlational measure, so it cannot tell us anything about directionality of the effects, meaning from this picture here, we don't know whether this connectivity is increased from the thalamus to the cortex or the other way around. So we needed to use a second method to be able to look at directionality. For that, we used dynamic causal modeling. Dynamic causal modeling has the advantage of revealing directionality between brain areas. It has, for the time being, the disadvantage that we need to focus on specific brain regions. We're not looking at the whole brain here, but really looking at regions of interest. People are working on overcoming this limitation right now, but here we had to focus on 4 brain regions, which we first of all knew are key for the thalamic filter model, and also know that they are implicated in the effects of psychedelic substances. So basically knowing that they are modulated when we administer LSD in that specific case. So we came up with 4 brain regions: the ventral striatum, the thalamus, of course, a region of interest in the temporal cortex, and the posterior cingulate cortex. And what we saw here is, again, warm colors, or red in this case, increased connectivity, blue, decreased connectivity. We do indeed see that there is increased connectivity from the thalamus to certain parts of the cortex. In our model here, the posterior cingulate cortex, but kind of similar like in our functional connectivity data, we see that this is not the whole story. There's also decreased connectivity from the thalamus to other brain areas, which is, for example, the temporal cortex here. So at least partially replicating or partially corroborating the predictions of the thalamic filter model. So seems like thalamic connectivity has something to do with the effects of our psychedelics. But of course, we didn't want to stop there. We also wanted to know what's going on in the rest of the brain. And for that, we used another method called global brain connectivity, which is investigating the connectivity of each voxel with each other voxel of the brain. So it's a data-driven method, doesn't require to get any seed regions or any regions of interest. The data-driven measure from each voxel to each other voxel of the brain. And what you can see here, I mean, even just by eye, you can see that LSD and psilocybin are inducing very, very similar effects. This one, psilocybin looks a little bit more scattered. The reason for that is because we used a different smoothing kernel here, but the effects are very, very similar. And what it does is that we see hyperconnectivity, increased connectivity between these sensory motor brain regions of the brain. And at the same time, we see a disintegration or hypoconnectivity of the associative brain regions in the brain, like the frontal cortex here, for example, and the default mode network here. So what does that mean? Well, first of all, to bring these different results together, we have seen that thalamic filtering is changed under the influence of a psychedelic, with more information potentially being passed through to sensory brain regions. And these sensory brain regions are then highly connected with each other and the rest of the brain, whereas the associative brain regions are disintegrated under the influence of a psychedelic. And that could mean that because we are integrating information differently, this could lead to these visual alterations that our participants are describing. And at the same time, it could also have clinical implications, because if we are integrating information differently, if we're bringing it together in a different way, that could potentially also give us a window of opportunity where we can break out of rigid thinking patterns that a lot of the patients are suffering from, where we can come up with new ideas and find creative solutions for potential problems which are associated with the disease itself. So I want to show you a couple of more results before we move on to the next part. So we did not only scan people during the peak effects, at least with psilocybin, we also scanned them 20 minutes, 40 minutes, and 70 minutes after administration. And what you can see here, it's very obvious when you're looking at the thresholded maps here, is that the changes in connectivity start in the occipital cortex, which is very much aligned with what our participants are reporting. So usually the first symptoms they notice are that the floor starts moving or the walls start moving, things like that. So very much aligned with subjective effects. Then 40 minutes, which is T2 here, 40 minutes later, we see that it's starting to not only be the visual cortex, but we get significant changes also in the associative brain regions. And then 70 minutes after administration, we basically see this full-blown pattern of heightened connectivity, increased connectivity between sensory brain regions, which is not counterbalanced by the integration of associative brain regions. Last result that I want to, no, not the last, but almost the last result that I want to show here is that I already told you that we were interested in the receptor pharmacology, so the serotonin to a receptor. And what we did here is we compared this pattern of changes in connectivity with cortical gene expression. Cortical gene expression maps derived from the Allen Human Brain Atlas. And here we saw that the pattern of gene expression for the serotonin 2A receptor is closely associated with the changes in functional connectivity that we measure with global brain connectivity. And another thing that we looked at is, can we predict the changes induced by, in this case, psilocybin, from the baseline connectivity of our participants. And what we see here is the changes at 70 minutes after administration. And indeed, there seems to be an association between baseline or placebo functional connectivity and the magnitude of changes induced by the substance. Okay, so this brings me to the second part of the talk, which is, well, now that we know that information is being processed differently in the brain and it's being sent differently from one brain region to the other under the influence of a psychedelic, what does that mean when the brain now interacts with its environment? And the first study that I want to show here is one on emotion processing. And this is a behavioral study where we again administered psilocybin plus Ketanserin, and placebo. And what we saw here is that psilocybin reduces the processing of negative information. And it's only negative information. It's not the case for positive or neutral information. So decreased processing of negative stimuli, again blocked by Ketanserin, so seems to be dependent on the serotonin 2A receptor. We did a similar paradigm also on the scanner, and we saw that psilocybin reduces again the reactivity towards negative stimuli in the amygdala. But we also know that the ecological validity of these kinds of tasks is sometimes not as satisfying as we'd like them to be. So we continued thinking about, well, what is a negative stimulus that psychiatric patients encounter in their everyday life? And what they sadly encounter a lot is social rejection and being excluded from their social environment. So we decided to run a pretty standard paradigm. It has been replicated quite a few times in the literature by now, which is called cyberball. And what happens during cyberball is, well, first of all, before the game starts, is that our participant meets Michael and Sarah in this case, and then enters the scanner under the influence of either placebo or psilocybin, and the participant plays a ball throwing game with Michael and Sarah. Now, after a while, Michael and Sarah stop playing the ball to our participant and exclude them from the game. And the beauty of this paradigm is really that it induces a feeling of being socially excluded. So it was fascinating to see that almost everyone who got out of the scanner was telling us, like, wow, that was actually really, really mean. Even though, you know, it's just a ball game, but still, it induces a robust feeling of being socially excluded. And as I said before, we're definitely not the first to do that. So the first study was conducted in healthy participants, and they found that being socially excluded from this game increases the bold signal in the anterior cingulate cortex, which is also why some people now name this activity in the anterior cingulate cortex the social pain signal. Importantly, this has also been done in patient populations, in this case, borderline personality disorder patients. And these patients show an increased reactivity, so increased bold signal in the anterior cingulate cortex. So what happens when we do the same thing under the influence of psilocybin? Well, of course, first of all, we asked our participants how excluded they felt. And we see a significant reduction in the feeling of being excluded under the influence of psilocybin compared to placebo. Now, of course, we are aware that our participants are under the influence of a psychoactive substance, right? So we need to check whether they were even aware of being socially excluded. So we asked them, for example, to guess how many ball throws they had received and asked many other control questions. And they were performing just the same as on placebo on these control questions. So the effect was really very specific on this feeling of being excluded. And when we looked at our brain imaging data, we saw that this social pain signal, so the increase in bold signal in the anterior cingulate cortex, was reduced under the influence of psilocybin. Another thing that we were interested in, while we're still at the topic of social cognition, was empathy. And so we did a behavioral empathy task. And what we saw here is that psilocybin increases empathy, but it only increases emotional empathy. So it increases feeling with someone. We also tested for cognitive empathy. So how well can they identify emotions in other people's faces? And there was no change for cognitive empathy, which in a way is a good result for us, because it also means, given that there was no change, that they were able to perform the task. Again, and under the influence of a psychedelic, it's always good to have a control condition. And yeah, we did not see any decrease in cognitive empathy, meaning they were able to complete the task, which also gives us confidence that the increase in emotional empathy is true. And by now, this result has been replicated quite a few times, also with LSD. So yeah, there seems to be an increase acutely in emotional empathy. We also tested for moral decision making, did not find any impact on moral decision making here, at least acutely. We still don't have very little data on long-term effects. So these are all acute effects. It's of course possible that things like cognitive empathy or moral decision making are just not altered in an acute state, but might be something that develops over time. But at least in our studies, we unfortunately have not tested for that. And there's still very little studies out there which really look at the long-term effects. So knowing that psychedelics have pretty profound effects on social cognitive processing, we were also interested in social influence processing. And what we did here is, this is now again LSD. We had people again in the scanner, 3 conditions: placebo, Ketanserin plus LSD, and LSD. I just want to point out the LSD effect here. So what we saw when we don't challenge our participants too much, meaning that their opinion is not so different from a group opinion, we see indeed that our participants adapt more strongly to the group condition, to the group opinion, than under the influence of placebo. And that is kind of what we already knew from some small behavioral studies before, that psychedelics increase suggestibility. However, as so many times, it seems like things are a little bit more complex, because if we start challenging people and provide them with a group opinion that is very different from their own, so sorry, a high conflict condition, then we see that there is a significant difference. So it seems to be specific. So the suggestibility seems to be specific for conditions where people are not in a lot of conflict with the group opinion. And we also did that while we were doing fMRI scanning. And here we saw that these changes seem to be dependent on the bold signal on activity in the medial prefrontal cortex, and the fMRI data basically mimic what we've seen in our behavioral data. And we were also able to now look at 2 different time points: one time point when they receive the social feedback, and one time point when they make the decision that itself. And we found these changes in activity in the medial prefrontal cortex already when they receive the feedback. So social feedback processing seems to be changed under the influence of LSD. And the last slide I want to show here is another one where we're also trying to figure out the mechanisms underlying psychedelics. And what we did here is we did a mismatch negativity paradigm, a paradigm that we usually conduct in the auditory modality, meaning we present the same stimulus over and over again, and after a while, the stimulus changes, inducing a surprise response or prediction error that is usually reflected in an EEG component called the mismatch negativity. We did that again with psilocybin simultaneously with fMRI and EEG, but we changed it from auditory to tactile. So we placed electrodes on the forearms of our participants and stimulated them with various intensities. And what we see here is that the mismatch negativity that we expect and that we also see when under placebo is reduced under the influence of psilocybin, meaning that there is not such a big difference between the deviant, so the new stimulus, and the surprising stimulus, and the standard, the old stimulus. And also when we look at the EEG data, we can start to understand why this might be the case. So again, here we see the usual response that you expect when looking at deviant processing. So heightened response here in the frontal brain regions. But under the influence of psilocybin, the standard actually looks very similar to the deviant, meaning that it seems like under the influence of a psychedelic, all of these stimuli seem to be surprising in a way, seem to be novel in a way, which might be the result of a memory trace not being formed. It might be the result of increased thalamic processing, so increased or less filtering, like we've seen in our MRI data, or it might be the result of a reduced integration of incoming information. So to sum this part up, we are now starting to understand what psychedelics are doing in the brain and how they elicit this altered state of consciousness. There are, of course, still many open questions, but we're starting to get an idea of what is happening. But we still haven't figured out how all of this relates to the potential clinical mechanism of action of these substances. And the issue here is we still just don't know. So I can only make a few suggestions why these substances do have the clinical effect they seem to have. And one idea here would be that there might be long-term alterations in the serotonin to A receptor system. There might be induced neuroplasticity, which is an intriguing hypothesis, but unfortunately so far only shown in animal studies. There might be changes in neuroinflammation, again, another mechanism that we've so far observed in preclinical models. There might be changes in epigenetics. We have seen these alterations in brain networks, which I already talked a little bit about, that this idea of forming or having a window to form new ideas and creative problem solving might be helpful. There's still little data on long-term changes in brain connectivity, unfortunately, so far. We have seen, as I talked about, these alterations in reward or emotion processing. We're starting to get more data here, and it seems like there are long-lasting effects on amygdala reactivity, at least in healthy participants, that last for up to roughly a month, or well, let's say differently, we know that by 1 month they return back to baseline, but at least they seem to be there for at least a week. A lot of patients report increased insight into dysfunctional behavior. And we've seen these changes in social processing, and also participants report increases in social connectedness. And we know that patients suffering from psychiatric illnesses are socially isolated, are are socially withdrawn oftentimes. So this increase in social connectedness could, of course, have an impact on, first of all, the patient therapist relationship, but also help them to reconnect with social environment, which then again might provide support for the long-term treatment success. So to wrap it up, what I think we know, at least to a certain degree, is that we have this pharmacological component of psychedelics, which is changing our state of consciousness. But we also have a lot of non-pharmacological mechanisms at play here, just because our brain, as I said, is not an island, but is interacting with the environment, and it's processing stimuli of the environment differently under the influence of a psychedelic. So psychedelic-assisted therapy, as it is conducted right now, can be understood as an interaction of pharmacological and non-pharmacological mechanisms. If these changes in brain connectivity really contribute to the clinical mechanism of action, then we need to think about the role of the therapist, and maybe the therapist can support finding new perspectives and support the creative problem solving. We also might have an opportunity for processing negative life events that might even extend after the acute effects, which again can be very beneficial for depressed patients, for trauma-related disorders who often have a history of negative life events. We have seen that there might be an opportunity for reconnecting with the social environment, which could also be actively supported in the treatment. And we might even have objective stratifying biomarkers. I've shown you this correlation between the baseline functional connectivity and the magnitude of changes induced by the substance. Of course, these are all healthy participants, as in most of the studies that I've shown today, in all of them actually. So all of that needs to be investigated, needs to be tested in clinical populations. But I think we should really take these questions and these mechanisms into account and do the work and figure out what it is exactly that is helpful for patients, because eventually we, of course, all share the same goal to eventually provide patients with the best psychedelic-assisted therapy possible. And I think that is only possible if we understand the neurobiology of these substances. And with that, I want to thank everyone involved, and of course, you for your attention, and I'm very much looking forward to any questions.